Good morning. Thank you, Gianfranco, for your introduction and the wonderful hospitality. I'm pleased and honored to be with the Accademia and contribute to the day of crystallography, of course with focus on my own topic of crystallography, which is biological structure determinations. Now I would like to start with a few slides of the history of post-diffraction crystallography, using the terminology of Professor Ferraris, and remind you on the Laue Centennial, which we commemorated 2 years ago. This year is 2014, the year of crystallography. Now what you see on this slide, I hope you do, is the first photograph hand-signed by Max von Laue of an X-ray diffraction of a crystal. So it's a miserable, ugly, smeared diffraction pattern, but it meant a revolution. In many respects, it was the foundation of crystallography as an analytical tool for chemistry, enabling to see molecules at atomic resolution, small ones first and then the big protein molecules later. It was a revolution because it showed that X-rays are electromagnetic radiation. Now, the X-rays had been discovered earlier, you know, but it was unknown what they really were. And it proved that crystals are regular lattices that are able to diffract electromagnetic radiation. Of course, it defined the dimensions of these lattices and the wavelengths of the X-rays, so on and so forth. And as I said, it revolutionized chemistry, biochemistry, material science. So all these many fields for which we use crystallography right now. Now this was Laue's publication in 1912 and I'm proud to say that, in the Berichte der Bayerischen Akademie der Wissenschaften. So a journal nobody reads, but it's led to a Nobel Prize 2 years later, so it's obviously not the impact factor. Now, impact factors at that time were not defined really, but you see, what is really important was clearly recognized very, very early. Now, this is Laue, and showing to Count Bernadotte, who is the founder of the Lindau Nobel laureate meetings, which are yearly events, and they take place again next week. I usually do find Italian students there. So it's an assembly of about 20 or 30 laureates with about 500 to 700 students which come there from all over the world. It's a wonderful meeting in Lindau, an island in the Bodensee, in the Lake of Constance, beautiful, close to the Swiss and Austrian mountains. So I just can recommend to the students and their mentors to do everything to find a place there. There are good chances. Now, Lauer showed to Count Bernadotte a crystal lattice. These are, as I see them, the founders of X-ray diffraction. There's Röntgen, this is Lauer. They're original instruments which you see on display in Munich in the Deutsche Museum. If you visit Munich, then don't miss a visit at the Deutsche Museum. It is great to be close to the instruments that changed the world in a sense. Now this is Röntgen's X-ray apparatus. This is Lauer's version. Now Lauer's experiment was spread very quickly, and rumors went to England, where father and son. Now, Professor Ferrari showed father Breck. This is the son, the founder of chemical crystallography, I would say. Now, these two scientists immediately grasped the importance of Lauer's discovery as a chemical tool to determine crystal structures, simple ones first, rock salt. Founded a school then later in Cambridge, England, where Max Perutz worked, who is the father of protein crystallography. He found a way to decipher the complex diffraction patterns of proteins. Now I come to biological crystallography, which began in the 60s. I, as a young postdoc coming from organic molecule crystallography, entered the field in the late 60s. So these are the numbers of determined protein structures close to zero for a long period of time, just less than a handful, until 1990. Then we see this exponential growth. The graph ends at 2008. It still goes on at a somewhat slower pace, simply because we know almost every basic protein fold, and because many of the structures that are determined now are worked on in pharma industry, and they are kept in their drawers, thousands, 10,000s of them, and I know about what is happening there in pharma industry, because they use protein structures as a tool for drug design and development, and there may be some time for me to mention that at the end of my presentation. So why this exponential growth? Because of the technical advances and the fact that in order to understand, oh, I'm sorry, it's somehow cut off here at the edge. Now, why is this so? Don't know. And applications in medicine. Right, there was a problem in the 80s, 90s for membrane proteins, which were difficult to prepare in clean, pure form. On the other hand, membrane proteins are most important drug receptors, because they control many physiological processes in cells, and they were thought to be not crystallizable. So my two colleagues, my former doctoral student Eisenhofer and a colleague Hartmut Michel and I thought we should embark on structure determination of a membrane protein, and we were lucky to take the right system, which was the photosynthetic protein in photosynthetic purple bacterium, Rhodopseudomonas viridis, gave unexpectedly beautiful crystals and a well-resolved high-resolution diffraction pattern. This was the structure which we published in '85. 4 components, 4 subunits, of which the middle ones, the 2 middle subunits, are embedded in the membrane, and they carry the chlorophyll. So this is where light is converted into an electric current. And on the basis of that structure, of course, physicists then did their calculations of the spectral properties of the chlorophyll, the electron transfer rates. So all of that, a fascinating period for both the structural biologists and the physicists to understand the fastest chemical reaction known at that time. So when light energy light is absorbed by a pair of chlorophylls that is in the center of this complex, then that pair ejects an electron which travels down a chain of chlorophylls until it reduces a quinone. This is the primary step in photosynthesis on which all higher life is based, as you know. Now, there was relatively slow progress from there on, but now we read about membrane protein crystal structures almost every second week in top journals because of their importance in physiology and drug design. Nobel Prizes have been given for work in that area. The last one in Nobel Prize in Chemistry 2012 for a G-protein coupled receptor. So obviously the community has learned how to deal with membrane proteins. Now a few words about the technical progress. This is, we have recombinant proteins and we have crystallization robots. So what we did by hand in the 60s, 70s, 80s, 90s even, is now done with natural proteins at that time, is now done with recombinant proteins. So we can make rare proteins in bacteria, in yeast, in human cells. And we need only tiny amounts because we have robots that set up thousands of crystallization trials in a short time. We have the huge X-ray generators, the synchrotrons. So we took advantage of what the physicists have built for their purposes. Now this is the Hamburg instrument on which we worked early on. The small ring, the Doris ring, were actually the first experiments of X-ray diffraction. Synchrotron radiations were done. Now, a very much larger and much more powerful, the Petra ring has been reconstructed, redesigned to be used as an X-ray machine, and there are dozens of synchrotrons for that in use. We're lucky in Europe to have so many and have relatively easy access to synchrotron radiation. This is an experiment. Diffraction of protein crystal recorded on a synchrotron. So what we have to do is we rotate the crystal in front of the X-ray beam and record within fractions of a second a single diffraction picture evaluated at the same time, very fast detectors, very fast computers and algorithms, so that after a few minutes we have a three-dimensional data set collected with sometimes 1 million of intensity data. These are the raw data that we need in order to reconstruct then a protein structure from its crystalline form. So enormous progress. This, by the way, were the models as we built them in the late 60s, early 70s. Metal parts. This was my work in the late 60s on an insect protein. This is a historical slide with David Phillips, who determined the first enzyme, lysozyme, crystal structure. This is how presentations, scientific presentations, were honored in that period of time. I mean, this is the Royal Institution. It's an auditorium, certainly not as nice as the Accademia here, but look at the audience, ladies in evening gowns, gentlemen in tails. The lecturer, David, in tails, he shows the essential components. There is the final model, so a wire and screw model of lysozymes. This is a crystal lattice. And the unfolded polypeptide chain hangs from the ceiling. So this is how a polypeptide emerges from the ribosome and then it folds up. Now we build protein models in the electron density using computer graphics. Now the first of these machines were available and the first program was made by Alvin Johns when he was in Martinsried in the late 70s. And the programs have developed such that even the interpretation of a good electron density map in terms of the amino acid sequence can be made automatically. In most cases, the density is not of that quality, and it needs interference of an experienced worker, but sometimes it is can be achieved automatically. Now, these are the techniques in structural biology that we now use the X-ray diffraction. I'm focusing on electron microscopy, an enormous progress in that field, again by the technical advances, mainly the detectors, but also computation. So we now see, again, if we open top journals, we almost every week see a medium resolution EM structure of these very large biological complexes. There is a very fruitful interplay between X-ray diffraction and electron microscopy because we get high resolution, atomic resolution views of a protein that may be a component in a large complex, and then we model the somewhat diffuse EM structure, that is at 5 to 7 angstrom resolution. We model this with the help of the high-resolution X-ray structure. This is a technique which is extremely powerful. The advantage, of course, of EM is that they work with single molecules. They do not need crystals. That is NMR. I have no time to talk about that. It has its special virtues, in particular because there's a spectroscopic technique. It gives us information on dynamics. Now we study proteins because the DNA sequences do not give us the information that we need on life phenomena, and this is particularly nicely shown here. This is a mature butterfly, this is its larvae, they do have the same DNA. So how comes? In order to understand that, we have to know which proteins are expressed in these different developmental stages and how they look like and how they interact. This is why we need protein structure determination. A few slides on what we know, how proteins are made. They are made on the ribosome as an unfolded chain, and then they fold up in a still somewhat mysterious way. This is the holy grail of structural biology, the solution of the folding mystery. Nature knows how it happens, because it happens. We don't know, or we don't know sufficiently that we can accurately predict from the sequence the three-dimensional structure, but we know that the three-dimensional structure is determined from the sequence. So we should be able to understand that sometime. Next generation, or the generation further on, I don't know. Now, the models that we produce are metaphors. This is a large protein that I worked on the last 10 years, the so-called proteasome, consisting of 28 subunits. This is an all-atom representation. We determine the position of all these about almost 100,000 atoms, or what have you, 50,000 atoms. If this is not understandable. So we have to reduce the presentation by just showing the polypeptide chain fold, or just showing the surface, or just showing the arrangement, the architecture, the arrangement of these subunits. We use metaphors for describing substructures, which we often see. So these we call propeller structures. When a polypeptide chain folds into these blades, one after the other, 6-bladed propeller, 5-bladed propeller, we call them barrels, and it's quite a fascinating resemblance. Also concerning the symmetries, this is 8-fold symmetry in the Castel del Monte in Apulia. We also have 8-fold symmetry in these barrel structures. We have symmetrical subunit arrangements. Now, proteins like to be together. Homo oligomers, hetero oligomers, and we have symmetries, 2-fold, 3, 4, 5, 6, 7, and even higher rotational folds. We have dihedral symmetry when a rotation axis is intersected by a perpendicular diode axis. Then we get this closed arrangements. We get shells. Well, I inserted this slide, which shows the symmetry that one finds in the cell walls of diatoms. Equally beautiful. And of course, the basis of this symmetry in these silica cell walls is the proteins, the proteins that make these cell walls. They must have that kind of symmetry as well, which is then reflected in the next level of the building up of an organism. Proteins are made on the ribosome, they fold, and they are sensitive, they need to be degraded. Again, crystallography helped to understand the ribosome, so the protein synthesis machine. Ribosomes walk along the message which is transcribed from the genes, messenger RNA, and then step by step synthesize the proteins, which are spontaneously folding up. This is, as I mentioned already, the mystery. We don't understand how that happens. And many are working on this problem, protein folding. There is progress, there's no doubt, but not enough in order to reliably predict protein structures from the sequence, although we know that the sequence determines the protein structure. Now, protein folding is sometimes not fast enough. You see, if an unfolded protein is around in the cell, then there is the danger that it aggregates with other components, other molecules in the cell. And then, of course, it cannot find its proper native structure, remains unfunctional. Now, there are folding helpers, folding catalysts in cells that essentially do the following. These very large containers, they essentially enclose an unfolded or not yet correctly folded protein inside a large container, so that it is isolated from the rest of the cell and has some more time to fold. Beautiful cage-like molecules. This was the first one that had been crystallographically analyzed, highly symmetrical. This is a bacterial protein folding catalyst called CROEL. We analyzed the eukaryotic protein folding catalyst called CCT, or in archaea, the thermosome. Now, there is a relationship which you see only when you look at the structure of these individual subunits. You find a relationship between the bacterial and the eukaryotic subunit structure. This is the American version, a bullet. This is the European version, a football. Looks nicer, I should say. But they do the same thing. They are cages, enclose an improperly folded protein, and by using some energy, ATP, which leads to some motions of these subunits, they give the enclosed substrate protein just more time to find its proper fold. Protein degradation. So what has been synthesized before in the ribosome, when damaged, has to be removed from the cell. It's non-functional, waste. This is a very essential process in cells, waste cleaning. It's a very essential process, as we know, not only in cells, but in all human communities as well. So we have many proteases which do that. Not just waste cleaning, this is one aspect of what proteases do, but also they are used for regulatory purposes, not by trimming a precursor protein, a pro-enzyme. Cleave off the pro part. The pro-enzyme is enzymatically inactive. For instance, the digestive enzymes, which are made in the pancreas, have to be synthesized in an inactive form, otherwise they would digest the pancreas. So they are secreted in an inactive form, and then in the digestive tract, they have to be activated by cleaving off the pro part. Therefore, we do have so many different proteases for these various physiological functions. They are dangerous, and they are not only regulation of protease activity by making inactive precursors, but there are also natural inhibitors of proteolytic activity. In the first one we analyzed in the early 70s. This is trypsin, the digestive enzyme. This is its natural inhibitor, forming a very tight complex. So this was, at that time, the first example where we had high-resolution, accurate structures of the individual components and of the complex to find out the basis of protein, strong protein-protein interaction. So we analyzed many of them. Some work like a bread slicer. This is a huge hexameric protease. This is an individual subunit. The movie shows proteins and evolution. Now, we know that we are related to bats simply by looking at the skeleton. This is a bat skeleton enlarged 200-fold. This is a very easy, understandable indication looking at skeletons, looking at the bones. This is a text by Charles Darwin. And as it is not completely shown here, what are they doing? So let me read Darwin's text. We must, however, acknowledge that man, with all his noble qualities, still bears in his bodily frame the indelible stamp of lowly origin. So this is a wonderful phrase of evolution. Now, looking at skeletons, that's easy. But when we would like to compare insects and whales, these have no skeleton. So the first structure that I determined in the late 60s was that of an insect rat protein, insect erythrochlorin. And to the surprise of many, we found that a low-resolution picture, that it has a fold like sperm whale myoglobin, which was the first protein structure determined by Kendrew and Perutz in Cambridge. So at the protein level, we see the relationship, or we see the relationship in hundreds of thousands of examples. Nowadays, of course, what we compare is gene sequences and protein sequences. But at that time, in the 60s and the 70s, there were no gene sequences and there were no protein sequences. But we looked at the three-dimensional structure and found the relationship. This is another example. This is a bacterial proteasome. I come back to that later. And this is a eukaryotic proteasome that we found in yeast and in mice and in humans. They are clearly related, no doubt. Showing, by the way, evolution, because the architecture of these two forms of proteasome is the same. It's a cylinder made out of 28 subunits. But the chemistry of the bacterial proteasome is much simpler. It consists of just two chemically different subunits. And it has 7 alpha, 7 beta, 7 beta, 7 alpha. Now the eukaryotic proteasome has all the alphas different and all the betas different. So it's a great example of how evolution works, maintaining the basic construct, varying it so to generate specificity. Now I see that time passes by, Gianfranco. Then I should speed up and skip a number of things I have on these slides to share. Photosynthesis, this is the work that led to a Nobel Prize in the 88. Photosynthesis, the basic process on which all higher life rests, seen from space in winter, snow, spring, chlorophyll color, summer, and autumn. These are the basic light reactions. Sunlight makes, with the help of the energy of sunlight, sugar is made, converting carbon dioxide and water into sugar and producing oxygen at the same time. So this is the basis of all higher life. So in plants, we do have the chloroplasts. We do know the basic components of photosynthesis in three-dimensional structure, meanwhile, but the beginning of that was our work by protein crystallography in the early 80s. There are two main components in photosynthesis. One component that collects light, so it's like a mirror, a waveguide, and then the photocell. Now, these are the cyanobacterial light collectors, huge organelles sitting on the membrane close to the photosystem, to the photocell. And this is an electron micrograph. It shows the fragility and the diversity when you isolate them. There's no way to crystallize these. But you can isolate the individual components, crystallize them, and look at their high-resolution crystal structures and find out how the system in principle works. I mean, if I speak of mirrors, then, of course, in biology, there's no glass, there's no metal, but there's just proteins and cofactors. And the question is, how can efficient light collection and energy transduction occur with biological material, and the basis of an understanding was by structure determination that we did, and then the physicists started to do calculations on the structures. Perhaps one simply understandable aspect of this system is that the outer components do have many more chromophores than the inner components, which means that the light is concentrated. So it acts like a focusing device simply by that means. Now this is the reaction center that I mentioned already. The first membrane protein that was analyzed, and the biological photocell. Again, evolution. Here I mention Jim Barber, where Gianfranco is associated with the Imperial College. A wonderful example of evolution again. Now this is the bacterial reaction center. This is photosystem 2, much more complex. 19 subunits, 4 subunits. But the heart of the system is identical. This is where the chlorophyll is bound. This is where light is converted into an electric current. This is maintained to atomic resolution. Well, I think I have to, given just a few more minutes, skip all of this. Just say use of proteins and in medicine, protein structure information, medicine and plant protection. What do we do in this simple slide? This is, by the way, a protein we worked on together with pharma for a long time, thrombin, the central clotting enzyme. Its apo structure, substrate binding site, modeling of a substrate of a ligand in here, synthesis, and then checking of our design idea and modifying the ligand. So this is what happens in pharma, in drug design and drug development in thousands of laboratories around the world. We can have a look at what nature does in this specific case, because nature, these uses has to, in nature has to inhibit blood coagulation in these blood-sucking animals. This is the leech. If it eats blood, then if that would coagulate, it could not digest, it would die. So it's a bag, the leech is a bag full of coagulation inhibitors, and we can learn from the structures of these coagulation inhibitors, and actually this has been done. So the main thrombin inhibitor in the leech is an approved anticoagulant drug, and also variants have been made on the basis of what we saw in the 3D structure. So one can use this part, combine it with a chemical tag, and have a drug that is called Hirulog and is also in clinic. Well, I have to skip all these wonderful things about the proteasome, my work, but perhaps show the way of academic discovery of a new drug-like molecule. We collaborated with a plant biologist who looked at a bean plant disease caused by a bacterial pathogen. And they found that this pathogen needs a virulence factor. Without that virulence factor, it's not pathogenic. They isolated it, and we found out that what it does, this is the chemical structure of it, and we found out that what it does, it inhibits the plant proteasome and kills the bean plant in that way. Now, the chemical structure now of this was synthesized and is now a basis for the evolution of many variants, which again are in the drug development process as a human a strategy for human cancers. Sorry to skip that, but would like to show at the end of a few slides on the foundation of business. Now, I told you in the last few examples that structural biology and the determination of protein structures of physiologically and pathophysiologically important proteins, that this helps to develop new medicaments. So this is a process that is well established in pharma industry. And, all the large pharma companies, but also crop science companies, do have structural biology for that purpose. But there is place for specialized small and medium-sized companies who just offer this kind of services to the large companies. And it turns out that this process seems to be becoming too expensive for big pharma, so they reduce their activities, sometimes in a very rude way. AstraZeneca just dismissed 200 people working in structural biology. So this is water on the mill of the specialized small companies. And one of them I helped to found 15 years ago, which does quite well. They have their own their own facilities in Munich. And what they offer to customers is all the steps of the process of drug design and drug development. That is, they make an interesting drug target protein. They even synthesize drugs. They crystallize and determine the crystal structures and give that to big pharma to develop further. So they do have already a very large collection of interesting proteins on the shelf. So of these drug targets, they do have protocols for making the protein, for crystallizing it, determine the crystal structure, and they even have protein crystals in the fridge. So if a customer comes and would like to know how his compound binds in order to develop it further, that information can be provided in a few weeks' case, because all the process of structure determination has been established. Very important for big pharma to have an answer quick. This is the other company I co-founded some years ago, which works on a new therapeutic protein for autoimmune diseases. Again, goes back to work that I did in the late 70s on antibody molecules. We continued that work on antibody receptors. Again, did the crystallography in the academic setting and found out how an antibody receptor binds to an antibody. And then we thought, can we make use of this information? There are many ways. Now, what one would like to do to interfere with an immune reaction, in particular an immune reaction against an autoantigen. These are quite disastrous diseases like arthritis, like systemic lupus, like multiple sclerosis, and there is no cure of this. So what happens there is that a patient has antibodies against its own constituents. And of course destroys muscle fibers in the case of multiple sclerosis, or thrombocytopenia. It destroys its own thrombocytes. So in that case, one would like to modulate the immune response, reduce the immune response. And here we thought we have a novel way how to do it, because if we interfere with the interaction of the receptor with the antibody, it's just the stem part of the antibody, then we can reduce the immune response. So what we did, we made soluble receptor as an antagonist for the cell-bound receptor, and that actually worked in a number of diseases, animal models, and then came I was fortunately not involved in the process of finding investors, finding money, but my colleagues were quite successful. So the soluble receptor is in various clinical phases, and it looks quite promising. This is Munich. As close to the Alps as Torino is, so this, but there is a special weather conditions which we call föhn, when the wind from the south, from Italy, comes very clean, very dry. Then we see the Alps like you see here in springtime. But I show this picture also because this is the old university, the Munich University. This is where Laue worked under the directorship of Sommerfeld, theoretical physics, and where Röntgen worked at the same time as professor of experimental physics. This is where the diffraction crystallography, Professor Ferraris, began 1912. Thank you.